My name is Justin MacCallum. I'm a junior fellow at Stony Brook University, and I'm here as an OpenMM visiting scholar. And my research is using physical modeling tools to understand and predict protein structures. I use OpenMM to do this research principally for two reasons. One is that it's very fast and efficient. It runs on GPUs, on computer graphics cards, which makes it incredibly fast. But I think more importantly is the flexibility. It's a library, and it allows me to combine different building blocks together in new ways and to try out new methods very, very quickly. So while I was here at Stanford, I worked on two different methods. One we call MeLD, modeling with limited data. And this is a method where we take sparse and unreliable data from experiment and bioinformatics, and we combine that with detailed physical models in a self-consistent way. And so the idea is that when the data is uncertain or inconsistent, we rely on the physical modeling to help decide what's correct and what's not. And at the same time, the experimental data helps to constrain where the simulation needs to search, and it makes the process of finding the structure much, much faster, which allows us to actually solve real problems that we wouldn't be able to use just using the computations alone. And so what we're looking at are problems where on its own the experimental data isn't powerful enough to get the structure, and the computation by itself wouldn't be powerful enough to get the structure. But by combining the two things together, you can actually get some very nice results. The other thing that I've worked on while I'm here is a method called the confinement method. And what this is for trying to figure out, given two structures, which one does a protein prefer and why? And so the key element is getting some mechanistic insight into, why the parts of the protein like this structure or that structure. And the ultimate goal is to be able to understand conformational change in proteins and what drives that. Which is very important in many proteins that undergo conformational changes during their active functional life cycle.